All right, so welcome to the biology practical session. So if you want to be good at this biology practical session, you have to practice something known as the scientific method. So the scientific method is just a detailed procedure that scientists use to make sense of the natural world. So what it involves is that you first have to collect data. You have to, first of all, you actually have to define the problem, you make observations, you carry out an experiment, you collect data, and then you make conclusions. So the biggest form of advice that I can advise you to do when you're doing any form of practical is to actually write what you see. Do not write what your friends are writing. Just focus on writing the results that you are actually seeing, because you don't know the reagents that you are going to be given. So don't go in there and memorizing procedures. Learn how to carry out the procedure, but do not memorize the type of results that you're going to predict. First, carry out that experiment. Then, if the observations are in line with the procedure, that's when you can draw conclusions. So in this part one, so I'm going to split this biology practical series into three parts. The first part will be the food test, as you can see on the screen, and the next part will be introduction to scientific drawing, where you are given maybe a leaf or anything of that sort, and you have to, draw what you see and be able to calculate the magnification from that using a specified formula. Then the third type of experiment that we're going to do will be in genetics, where we're going to look at things known as maybe modes of inheritance, so complete dominance, incomplete dominance, and codominance, because you might be given maybe a black and white leaf, and you'll be told to determine what type of inheritance is observed. So let's start off with food tests. And every time we're talking about food tests, really in our ECZ syllabus, the only food test that they are going to tell you to do will be carbohydrates and proteins, because basically that's what we can afford. So carbohydrates and proteins. And when we're talking about carbohydrates, there are two types of carbohydrates that they want us to test. The first one is polysaccharides. Polysaccharides, this is where you use starch, and the reagent that you use is iodine. Then the other class of carbohydrates that they want you to test are your monosaccharides. We're going to call these monosaccharides reducing sugars. So it can be glucose, it can be fructose, it can be galactose. And the reagent that you use here is Benedict solution. And the last type of food nutrient that they want you to test is proteins. And the reagent that you use here is a burette reagent. So under all these types of principles, you see, it's under all these types of tests, it's very important that you first know the principle behind these tests. You have to know how to fill in your table, because you're going to be given a table and you'll be told to write down the method that you see. And after writing down the method, you have to be able to make observations and draw a conclusion from what you see. So pay particular attention when we talk about the method for test for proteins using burette reagent. When we talk about the principle of the iodine test for starch, for polysaccharides. And when we talk about the principle of the test for reducing sugars using the Benedict's reaction. All right, so let's start off with a question, a hypothetical question that I created for your own understanding. So here, in an exam scenario, you'll be given three solutions in various quantities, probably in three different test tubes. And you'll be required to carry out maybe just one test, or sometimes you'll be required to carry out all the food tests in those three solutions that you've been given. So this hypothetical test states that you are provided with the sample solutions. So we have got three sample solutions in various test tubes, test tube X, Y, and Z. So the test tube labeled A, X, Y, and Z, they contain different solutions. So you don't know what type of solution it could be. It could be maybe a yolk solution which can contain proteins. It could be some form of orange juice which can contain monosaccharides, or it could be a solution which has pounded groundnuts which can contain starch or polysaccharides. So the question says you have to carry out food tests for starch, proteins, and reducing sugars. And remember the scientific method I told you about. So if you want to answer this practical, you have to carry out the scientific method, like I have just said. Carry out the experiment first. Do not memorize the conclusions that you are going to draw. Your observations have to be in line with a valid conclusion, because you do not know the type of reagent that you might be given or what it contains. So it's very, very important to just take a deep breath and actually carry out the procedure well. So we're going to start off with our test for starch. And the first thing that I want you to understand is that starch is a polysaccharide. As such, just wrap this part. So starch is a polysaccharide. It means it's made up of many units of carbohydrates. So many monosaccharide repeating units. So a lot of monosaccharides joined together in a glycosidic bond forms a very long polysaccharide molecule. So this very long chain of carbohydrates, known as a polysaccharide, contains amylose repeating units. And this same amylose has the ability to combine with iodine. So if I can say, let me say amylose, which is the monosaccharide repeating units that are found in a polysaccharide like starch. So you have got amylose. Amylose combining with iodine creates an amylose iodine complex. So I've got your amylose plus iodine complex. So this is now the principle. So what this does is that it's going to absorb light at a specific wavelength. So this amylose iodine complex is going to absorb light at a particular wavelength, and it's going to reflect a wavelength of blue. So this blue-black color is what you see as a result of the absorption and emission spectrum of the amylose iodine complex. So if that food substance contains starch, after you add in your iodine solution, you're going to see a blue black color. So you just say blue black color. Sometimes it's deep blue, but you'll be safe to just say it's blue black. So what is the procedure? This is actually extremely important, and I really hope you can see this diagram very clearly. So here you have got your so this is similar to how uh, you know your exam is going to look like. You're going to have something that you're supposed to fill in. So here you're going to have your solution or your specimen. So our hypothetical example, we've got three types of specimen, X, Y, and Z. They won't give you the method, so you have to know the method by memory. And you know all these methods, you just have to know it's the rule of two. So all these methods that we're going to talk about, you have to add in 2 cubic centimeters of solution, 2 cubic centimeters of solution. We are going to have a dropper. So you just get a bit like 2 cubic centimeters of the solution. Uh, you can measure about 2 cubic centimeters using just, you know, this is your digit, your hand. So this length of your hand is approximately 2 mils or 2 cubic centimeters. So for the stats test, you get 2 cubic centimeters of the test reagent. So that's 2 cubic centimeters. And then you add in, you get your dropper, you add at least 4 drops of iodine. After adding 4 drops of iodine, you're going to get this blue-black color. And the presence of this blue-black color shows that starch is present. So again, you have to write your solution there. Take 2 cubic centimeters of the sample or the specimen, add 4 drops of iodine solution and shake. So you have to shake for you to form that iodine amylose complex. So you shake. If the solution turns yellow, it means starch is absent. What happens to solution Y here? The mixture turns blue-black, meaning starch was present. The mixture turned blue-black, meaning starch was present. So you are carrying out the experiment in real time, but you're reporting your results in past tense. So be very careful. So again, your test for starch, what are you doing? You're just adding 2 cubic centimeters. Please don't write square centimeters, it's cubic centimeters. So 2 cubic centimeters of the sample, you add in 4 to 3 drops of iodine solution. Presence of a blue-black color represents that starch is present. And don't forget your principle behind the test, because starch is made up of a lot of amylose repeating units. It has the ability to combine with iodine, forming the iodine amylose complex that is able to absorb light at a certain wavelength and emit blue color at another specific wavelength, so the blue wavelength. So this image here shows a positive result and a negative result of the starch test. So if you see this yellow color, it means there is absolutely no starch in your test sample. But if you see this blue-black color, it means that the starch was able to combine with the iodine to form that iodine starch complex that's able to emit this blue-black color. So this is a positive test for starch, and this is a negative test for starch. So let's move on to our second class of food molecule that we want to test, which is proteins. So proteins, every type of protein is made up of repeating units known as amino acids, and these amino acids can form small chains known as peptides. So what this burette reagent does is that it has the ability to react with these peptides. So burette reagent reacting with peptides gives off a certain color. So this reaction results in a color change, which is purple. And it's very important to note that burette reagent is actually blue. So if you add in burette reagent to your test sample, the color has to actually change to purple. If the solution turns blue and remains blue, it means this reaction did not occur. It means the solution has no peptides in it. So burette reagent has the ability to react with peptides, and the reaction forms this purple color. So let's look at our hypothetical solutions and we see what happens. And again, your method has to be very, very clear. So here you are adding, because burette reagent is also a liquid, and the sample that you'll be given is also a liquid, so you have to add equal amounts. So if you are going to add 2 cubic centimeters of sample plus 2 cubic centimeters of burette reagent, and you mix well. So take 2 cubic centimeters of the sample and add 2 cubic centimeters of burette reagent, and shake gently. Shake gently. So this is the procedure that they want you to write in your method column. Then let's look at these results. So here our solution X, this mixture turned blue. It means proteins were absent. But Y here, the mixture turned purple. It means proteins were present. It means the burette reagent was able to detect those peptides that small protein units. And then Z here, the mixture turned blue. It means proteins were absent. So let's look at a picture to see what's happening here. Okay, so this is exactly what's happening here. As you can see, forget this picture here. It's just showing the preparation of burette reagent. What I want you to observe here is this part. So a negative test is going to show that, okay, the solution is going to remain blue, but a positive result for protein, you're going to see this purple color here. So proteins were present, proteins were absent. So let's look at our last type of food test, known as reducing sugars. And reducing sugars are just simply mostly our monosaccharides. So these monosaccharides have got a free aldehyde group, which is an OH group, and a free what is it called? Ketone? Yeah, a free ketone group. So this free aldehyde and free ketone group have the ability to reduce burette reagents. And because burette reagent is going to be reduced, you are going to see certain color changes upon heating. So you get 2 cubic centimeters of sample, you add 2 cubic centimeters of burette reagent, and then you go to your Bunsen burner and you start heating gently, and you're going to see certain color changes. You're going to see the mixture going from blue to green to yellow to orange. Then if this is a very, very concentrated solution with a lot of reducing sugars, it's going to turn into a very bright orange color known as a brick red color. A very, very interesting color. So let's look at our method. So you take 2 cubic, again please, 2 cubic centimeters of sample, not 2 square centimeters. That means something different. So you take 2 cubic centimeters of your sample, you add 2 cubic centimeters of burette reagent, and you heat gently. Then you start seeing your observation. So our hypothetical solution here, so here X, the mixture turned from blue to green, then yellow, then finally orange, showing the presence of reducing sugars. And I must warn you, Examples of reducing sugars include glucose, fructose, galactose. Sucrose is a disaccharide. So sucrose is a non-reducing sugar. It does not have these free aldehyde or ketone groups. So this will give you a negative reaction. So the solution will just remain blue or green. So let's look at what's happening to why the solution remained blue. So reducing sugars were absent. Sample Z, the mixture remained blue, meaning reducing sugars were absent. So please, these procedures, you have to know them. If it's starch, you're adding 2 cubic centimeters of your sample into a test tube, then you're adding 4 drops of iodine. If it's burette reagent for proteins, you're adding 2 cubic centimeters of sample to 2 cubic centimeters of burette reagent, and then you observe that purple color. If it's reducing sugars, you're adding 2 cubic centimeters of sample, 2 cubic centimeters of Benedict solution, 2 cubic centimeters of Benedict, was I saying burette reagent here? Sorry, this is Benedict solution. Burette reagent is for protein, sorry about that. So you get 2 cubic centimeters of Benedict solution, and you heat gently and you observe the color changes. If you see, upon heating, if you see your color changes going from blue to green to yellow, then orange, finally a very bright brick red color, it means your reducing sugars are present in the solution. So here's a real life example. So here, if it's blue, if your solution remains blue, it means no reducing sugars are present. If it's somehow green, it means just traces of reducing sugars are present. Mostly your test result is going to look like this. So here you can just say reducing sugars are present if it's orange, or if it's brick red, you just also have to say reducing sugars are present. So our end goal is to have something that looks like this or something that looks like that. So orange color is acceptable. But if a lot of reducing sugars have been observed, no wonder I said it's very important for you to carry out the experiment. You say the solution finally turned this brick red color, this color here, showing huge amounts of reducing sugars. So I'm going to end here with part 1 of this video. Then I'm going to record part 2 on scientific drawing, which will take me like 5 minutes or so. Then the last one on genetics will take another 30 minutes. I'll see you in the next video. Let me just upload this one. Good luck in your biology practical, and I hope this helps.